Yeah, so hello, my name is Madeline McCoy. I am an undergrad student at the University of Florida. I'll kind of talk about why I'm here in comparison to a lot of other people who are doing higher graduate level projects. But I'm hoping to look at free-roaming dog disease ecology and kind of how that relates to conservation, especially that of lemurs. So about me, so I go to the University of Florida. I will be graduating with a zoology and microbiology degree this fall. I secured funding to get here through the Center for Undergraduate Research, and previous research I've done has been in veterinary toxicology. So a lot of my experience is in molecular work, and as you can imagine, that's very different from a field season in Madagascar. So I'm kind of going to talk about how I prepared myself and made a plan to collect molecular samples and kind of what I've been trying to do. In the future, I hope to maybe get a DVM PhD. I'm not quite sure, but really this has been about, I'll get into this, but restarting a disease ecology program with MDI. So I took a class with Dr. Kim Valenta, who was a co-founder of Mad Dog Initiative, which was, yes. Oh, sorry, Marley. Oh, thank you. Sorry. Who was a co-founder of Mad Dog Initiative, an NGO that does spay-neuter campaigns in areas bordering conservation zones for lemurs or other areas of wildlife importance. So I took her class, Primate Ecology, because of my minor in anthropology, and I wanted to do some type of international study in general because I feel very limited as a student in the US. And I was asking her about Madagascar, and she says, "Why don't you just come to Madagascar with me?" And I was like, I don't know if I could do that. I've never been outside the US. This was a process where I had to get my passport, and she introduced me to this set of samples. So these are samples from 2017 to 2019 that have just been sitting room temperature in Dr. Valenta's lab for the last few years. So these were under development by a previous disease ecologist in collaboration, and there are hundreds of blood smears, of desiccated fecal samples, and other samples of importance. And she said, if you can do anything with these samples, please have at it. So I have a lot of interest in parasitology and, of course, veterinary science. And I spoke with clinical pathologists and parasitologists at my university who found really no viability of this set of samples. So at that point in 2023, it was really like, you have to go to Madagascar and restart this program because it's really important to know kind of cross-species transmission in these conserved areas where, as I'll discuss, free-roaming dogs are a really important invasive predator for a lot of species. So this is really how I got into this, and this is really a pilot study of me figuring out field seasons and figuring out how to do molecular work here. So I'm going to kind of talk about that. So free-roaming dogs is kind of a newer term in reference to feral dogs. In the US, we really don't have these where they are dogs that are owned by people maybe or associated with individuals, but they kind of have free reign of the environment. And in a lot of cases, MDI has seen that they have gone and left for either days at a time, weeks at a time, where the owner really has no understanding of what they're doing and their behavior. And so previous studies have shown that there's direct predation on a lot of really important species that really should be highly conserved, like tenrecs and lemurs, as well as direct competition with Malagasy carnivores. So in co-occupant studies where these dogs are entering into these protected areas, these Malagasy carnivores are being outcompeted or directly interacting with the free-roaming dogs. And so they are no longer interacting in those areas. So that's really important when you have these free-roaming dog populations directly bordering protected areas, like we'll discuss in Andasibe. It's also important from a veterinary perspective to look at the population health and welfare of these dogs. So in a lot of cases, there's limited access to veterinary care, and so there's a lot of overpopulation of these dogs. Dogs can reproduce very quickly, and there's limited access to vaccines and to general health care. So understanding disease prevalence both as an intra-population for dogs to kind of understand that subpopulation dynamics, as well as how they're interacting with endemic species. It's going to be really important for conservation going forward, especially with MDI. So, zoonotic canid pathogens, which I should say I'm mainly going to focus on parasites, but it's important to kind of cover kind of the full range. So there are a lot of zoonotic pathogens that dogs can do, and as a generalist species where they travel with humans everywhere and they will act as a generalist predator in those areas, they interact with a lot of different animals. So in terms of bacteria, you can get things like leptospirosis, salmonella, pasteurella, and then parasites, there are a wide range. If you go into GI, there's things like hookworm, Giardia, Cryptosporidium, and then you also have blood parasites like Babesia, Diomedes, kind of the list goes on and on. And then you have viral pathogens such as rabies, and rabies is a really big humanitarian concern, especially because in 2016 there was a paper published that referenced a direct interaction between FUSA and dogs that then possibly resulted in a positive rabies. So we have a positive rabies and we have direct dog FUSA interactions. So this kind of like full story kind of comes together to say that this could be a real issue going forward. We also have another study in 2019 where Diomedes was found, which is the direct species responsible for canine heartworm, was found in wild lemurs, specifically. Actually, I'm not quite sure. I was going to say Microcebus, but I wasn't quite sure. And this was done by the previous collaborator with MDI. So this population of dogs is directly an example of a sentinel species, which is a species that has a disease and is a representative of the disease in a total population and can be spread to other more vulnerable populations. So in disease ecology, when you bring in any new organism to an environment, whatever pathogens that this has is going to leave the rest of this endemic population is going to be very vulnerable to whatever this invasive predator has. So because dogs are doing direct predation as well as direct interactions with a lot of species, there's a lot of cases for transmission. There's also the case of vectors. So mosquitoes and ectoparasites are at risk, both as parasites and as transmitting bacterial and viral infections. So in general, dogs are a really big risk for conserved populations and in general as populations themselves. I believe that makes sense. So knowing this from literature and from Dr. Valenta is very different than coming here. So I made a collection plan that was based on the MDI spay-neuter campaign, and I made a lot of plans for molecular preservation, but a lot of things are different here than I thought. But in terms of a collection plan, we had 3.5 weeks of a sterilization campaign around Andasibe. And so this is a picture I took from Google. It's my own screenshot, and that I had made for a grant for funding. So Andasibe is right here, and then we have this is a protected area, and so this is one of the closest place that we went to the protected area, so a low density population with dogs that are directly interacting with conserved populations. So in the spay-neuter campaign, we're going all along the conserved area in an attempt to reduce the dog population, and I would be collecting samples with the Malagasy veterinary team and kind of try to assess general parasite diversity from a variety of samples. So one of the difficulties in assessing pathogen load would be a lack of a cold chain here. My understanding coming here is that I would have no access to refrigeration, especially not a -20 or a -80, which is usually standard for a lot of samples that you're going to receive, especially blood or fecal samples. So finding a way to preserve my samples, traditionally in like formalin or ethanol, was very important and was an interesting process going forward. Also, there was the possibility for Microcebus samples. in association with Caitlin Filla, a PhD student in Dr. Valenta's lab, that I could possibly use to do some analysis with my fecal samples. So my plan coming in was just to do pathogen diversity based off of a variety of samples, which I'm going to explain now. So based off of canine heartworm study in 2019 from Dr. Sarah Doty, my main focus would be Diamidis and its prevalence in this dog population, especially closer to Mantadi in Lake Mahatsara. Because of the difficulty with molecular analysis, I was really trying to find a way to do an in-field processing, which I actually did find in a SNAP-40X test, which is from a company called IDEX. So this is an in-field ELISA that can actually be at room temperature for 6 months. Usually it's refrigerated because of a lot of the reagents can be sensitive. But this was my plan that I made with parasitologist and Dr. Valenta to kind of get an immediate answer that maybe wouldn't be variable in transport. And so we have the SNAP test. I was also going to preserve the blood as best I could in anticoagulant, EDTA, for a further modified NOTS test. Diamidis antigen is the main thing that is going to be presented in the SNAP-40X test. And because it's only the female antigen for the microfilariae, it is not always going to be inaccurate. So you can have a lot of false negatives, and you can have a lot of false negatives which need to be verified by a visualization of microfilariae. So that's going to be my plan. We'll discuss that later. I also collected fecal samples. So gastrointestinal parasites are one of the most common that are assessed because fecal samples are really easy to get, kind of from lemurs and from dogs. And my plan is to do a DNA extraction as well as a direct parasite morphology with using a fecal flotation. This is as the added benefit of me getting microcebus or mouse lemur samples, so a direct comparison is possible. I also collected fur for some separate researchers at the University of Florida for isotope studies. Thought I should mention it, as well as directly collecting ectoparasites for direct morphology and hopefully molecular analysis down the road in the works. So I have some expected outcomes because this is a research study, but it's really still in the works. So I assume that there will be a high parasite density when you have a high density of dogs in an area. So this is not necessarily going to be true in places like Mahatsara, directly close to conservation areas. But I think that there may be a high prevalence of Diamidis because vector-borne parasites can be transferred by the prevalence of vectors, and wherever there are mosquitoes in hot conditions, it's pretty much everywhere. So I assumed I would find a lot of Diamidis, especially because there's really no way to do prevention in these areas. I assumed that there would be a high diversity in GI parasites because of the high diversity in diet from a lot of these dogs. Like even if you're living in a city, you're getting a variety of diet based off of past MDI and this field season's information. I also think that ectoparasites will be associated with dogs and not necessarily cross species, because I think that the direct species interactions are difficult to kind of transfer ectoparasites. And I hope that molecular data will kind of back up my morphological identification that I will show you going forward. So SNAP test is the main result that I have because it's an in-field test, and so I could get the results within 15 minutes. Out of 213 blood samples of canine samples, I only had 6 positive Diamidis samples. One of them was in Mahatsara, which is directly bordering that conserved area, and the rest of them was in Moramanga, which is a larger city nearby. This was quite surprising to me, considering that in the US, in shelter dog populations, you can have a 20 to 50% population with Diamidis in a very large scale. So it was really interesting to see that. We only had 1 positive Ehrlichia. Burgdorferi and Anaplasma are 2 other tick-borne diseases similar to Ehrlichia. Diamidis is transferred by mosquito. So these vector-borne diseases really, I assume, to be way more common than they were. So when you're reading a SNAP test, you kind of have 4 areas to look at based off of a positive test. So to see if a test is working, you have a positive control, which is basically a reaction between when you put a reagent to mix with the blood, it shows that the reagent has positives in it that make sure that you know that the test is working. And then if the blood has some other positive antigens or antibodies in it for these other pathogens, then they will show up. So I have some examples of that in the field where I'm taking pictures. So we have some Diamidas in different locations. Right here, it's this really faint blue dot, so it's kind of hard to see. And we have some Ehrlichia here. So no matter how faint this dot is, it means there is some amount of antigen or antibody of this pathogen in the sample. So we have to kind of assume that all of that is possible and hopefully do more molecular tests down the line. So this was quite interesting, and this is the main result that I have. But we also, in the future, I'm going to do a modified knot test that I referred to. So modified knot test is done with whole blood and is a concentration to look for microfilariae, which is the small worm that is Diamidas. So you can do this by lysing red blood cells and removing them from the sample and concentrating it down with centrifugation or with filtration. You dye this pellet and you search. The problem with a modified knots test is that it's very difficult to differentiate really similar microfilariae, and this was a lot of concerns with Dr. Zodi's paper as well of, is it really Diamidas or is it very similar microfilariae? So the way you tell, it's kind of hard to see as well, the way you tell Diamidas is this really tapered head on the microfilariae and the very tapered tail when you're looking at it on the slide. This is not Diamidas because it has a much blunter head, and because there was no picture, there's a lot of suspicion about that. So I'm hoping that I can take pictures and show my positive microfilariae if I find Diamidas, so that there's not as much debate about that. So yes, my fecal samples, because I'm looking for GI parasites and because shedding can be kind of difficult with cysts and trophozoites, I'm doing a DNA extraction to verify molecularly in the US. So I did 104 samples, including microcebus samples, and in a DNA extraction, your goal is to kind of extract all DNA and then specify which DNA you would like to amplify later for different species identification. So in this process, you basically lyse host cells, you bind DNA using different binding buffers and wash off all the things that you don't want. So in this molecular process, I spent a lot of time in Madeline lab. I did 104 samples, and I worked with, this is Caitlin Filla, and this is Anj, who's here with us today, to kind of process as much fecal as I could. And then afterwards, I was going to do a fecal flotation to do a morphological identification of any parasites I could find. My goal would be, if I have a morphological identification, that I could back this up with a molecular identification. So the way you do a fecal float is you have to create a specific gravity solution that is higher than water. So Sheether's solution, which was one that I could create here and I didn't have to bring from the US, is at 1.25 specific gravity. This allows eggs that are going to be lighter than a lot of debris inside of inside of feces to float to the top, as well as helminths, nematodes, and flukes. Adding centrifugation to this is really helpful rather than doing a passive bench top, and is the current procedure for that. And then you can create a reverse meniscus, put a slide cover on top of that, and then read it from there. So I have both canine and microcebus parasite examples. I also have not read them because we just did them this week, actually like yesterday, but I have a lot of suspicions of what we have here, possibly some hookworm, whipworm, some other types of nematodes that will be really interesting to identify going forward. In general, canine samples had what appeared to be a high diversity, but I have to go through and actually process that data. So that's mainly a qualitative assessment. In terms of lemur, because there's such a small sample, usually around 1 gram, which is less than the preferred 3 to 5 grams of the general procedure. There wasn't a lot of parasites per sample, which I assumed would happen. But in the ones that did have parasites, the parasite load seemed to be really high. So in the future, I would really like to do a quantification of that, which is a different procedure than just the regular qualitative flotation. These are some of the worms I found. They're quite interesting, and it'll be interesting to find a morphological ID for Microcebus-specific parasites, but that's my goal going forward. I also need to do an ectoparasite identification. This is an example of what I would like to do. I really mostly have fleas and lice. There's not as many ticks as I also assumed that would be here, but having a visualization of what these ectoparasites are and species identification is really helpful, especially if they are vectors for other diseases. So I would like to thank the entire MDI team for kind of letting me hang around and collect samples. It was mainly before a surgery, so it was really quick, and it was really nice of them to let me collect these samples. And I would also like to thank Caitlin and Madeline Lab and kind of everybody that's helped me along this journey. It's been extremely interesting, and I have a lot to learn going forward. So, thank you.